Introduce Dr. Naima Moustaid-Moussa. She's all-horn distinguished professor in nutritional sciences and director of the obesity research. Her PhD in endocrinology at the Sorbonne University in France and postdoctoral training at Harvard School of Public Health. Her lab studies the dietary and endocrine regulation of adipose tissue with the adipocyte renin-angiotensin. How bioactive compounds such as fish oil reduce white adipose tissue inflammation, activate browning, increase lifespan, and alleviate metabolic. Her work has been heavily funded by federal agencies such as NIH and NSF, as well as by foundation agencies. Actively involved in the teaching and mentoring of undergraduate and graduate students, postdoctoral researchers, and faculty. Has over 160 publications, including publications in gender equity, 4 published patents, and has received multiple awards. The most recent Pfizer Consumer Healthcare Nutritional Sciences Award and the Korean Nutrition Society Award. Fellow of the Obesity Society, fellow of the American Heart Association, member of the ASN Board of Directors, and serves on the NIH Human Studies of Diabetes and Obesity. So please join me in welcoming Dr. Naima Moustaid.
Thank you. Thank you very much for that introduction. And just like if I am not doing enough service, I was just appointed to the Board of Agriculture and Natural Resources of the National Academies to serve for a 3-year term. So I could not say no to that one, right? Yeah, but thank you very much for having me here. I usually like to move, but because I have to use the mouse, I'm here, but I hope you can all, see me. I'm very flexible how you want to do the questions. You can interrupt me anytime if you want to wait until the end, that's fine too. But I really appreciate the invitation. This is my first time to Colombia, and I've enjoyed so far all the meetings, very exciting. And I was telling some of you, Vicky and others, oh, maybe I should, now that we talked, I should have actually talked about this, not that. But so I'll give you an idea of what we're doing in our lab. And then, if anyone is interested in those, we can discuss as well. So we will talk about, of course, my passion is adipose tissue. You can see from the image, I love adipocytes. They're not all that bad. They have some good aspects. So that's why I'm still working on that since the 80s. But I will cover 2 aspects of the research we have done, one on the endocrine regulation or function of adipose tissue, and the other one, how we can use dietary components to alter adipose tissue. And for the endocrine function, I will primarily talk about the renin-angiotensin system, just summarize some of the work that we've done over the years. And then for the dietary approaches, I was going originally to talk just about the obesity and insulin resistance and potential role of uncoupled protein 1. But then I decided to add in the Alzheimer's story because we have some exciting, data that we got recently that I thought I would share. And maybe I hope to get also some of your feedback on some questions that we still have. Most importantly, I need to recognize really the people who did the work in my lab, and you wonder what the green and the red colors are. Shane here has red and green, and if I had expanded the topics, he could get all the colors. He's my lab manager. He's been with me since I moved to Texas Tech. He does ordering, training. So he's, a really very important person in the lab. So is everybody else, but he has a special place and role. And then the green circles actually are for those who worked on the fish oil. So Mandana Pahlavani actually was the one who had the idea after she saw our data on fish oil and inflammation. And she asked, how about brown fat? I was not interested in brown fat. So you students, you give us ideas that benefit everybody. So I love that. And from there, actually, we expanded it. And she was actually the reason, I think, why our grant was funded, because she had some, preliminary data that showed, what I'm going to show you later on, that really supported that idea. And the Alzheimer's study was done by Mahsa Yavari. She's actually finishing up. She's someone who came with clinical training, never touched a mouse. And then she bred the Alzheimer's mice and developed this study. It's a new project in our lab because I'm not expert in neuroscience. So she has done a lot working with our neuroscience collaborators and learning the studies. And then the red circles actually are for those who are working in the renin-angiotensin system. Of course, I have to mention Nishank Alopahana was one of my earlier grad students in Tennessee, and he works both on fish oil. He should have actually the yellow and the green and the red circle as well. He works on both fish oil aspects and the renin-angiotensin system. He's now at the University of Pennsylvania, and they just heard actually this week, it made my week. He got promoted to senior professor. That's the highest level of professorship you can get. And he was also chair of physiology at the School of Medicine. So those are the kind of things that really make me cry and excited and happy when my students do well. And Latara Malingam was postdoc in my lab and then research assistant professor. And then she got a tenure track position at Syracuse University. And Kalhara Minik Dawela also worked on the renin-angiotensin system. I'm going to mention some of his work as well. And half of our lab, most of these students, except for her, are undergrad students. We have usually 12 to 15 undergrad students in our lab. It's a requirement for every grad student, every postdoc to have at least 1 to 3 undergrad students that they mentor, because that's, I think, just an invaluable experience. And then I have long-term collaborators at Texas Tech University, at the University of Houston, mostly for the genomic studies that we do. Marshall University with Brazil, we have developed a really nice collaboration with William Fistoshi at the University of Sao Paulo, and also with Qatar Computer Research Institute for Bioinformatics. So I'm spending time on this slide because it's really very important. What I'm showing, most of it I didn't do, I just trained on specific techniques, but it's really the people in the lab who make this work. So our work was funded by NIH for the fish oil and UCP study that I will show. Angiotensin work was funded by the American Heart and by USDA as well. I'm not going to talk about this work, but we got recently some funding from industry on the to look at some beef proteins. So I appreciate all the funding that really helps with what we do. So I think you've all seen these maps and how obesity has risen. So I'm not going to spend a lot of time on that, but I want to make a couple of points. If you really look at Calvary, the maps, you see differences. And this survey from 2017 to 2019, you can see that there are fewer states that are very red in terms of obesity, that have obesity rates over than 35, and these are non-Hispanic whites. But then if you look at the Hispanic adults, look at the picture, it's much darker, and they look at the non-Hispanic Black adults. I don't work in the area of disparities, and I cannot model it in mice, although we have different color, mice of colors too, but we cannot really model that. So I'm telling you, those who are working in the disparities area, that we need really to pay attention to that there are differences in diseases. And I think most of you know this figure. I use it a lot in teaching. This is an infographic that was developed by the Obesity Society back in 2015, 2016. And while it does not show what are the causes of obesity, this is just a summary of studies that have been done that show that there are many factors that can contribute to energy storage, and those were divided into inside the person or biological drivers of obesity, age-related changes. I can feel it. Chronic inflammation is another thing, thermogenesis, gut microbiome. And then you have on the other hand, external factors, environmental factors, maternal stress. Some of these, of course, are debated, low socioeconomic status. So the chemicals, toxins. So there are a lot of factors inside and outside the person that can, drive obesity. But what we're really interested in is this chronic inflammation, is the role of adipose tissue. And I know you have a lot of background in this, so I'm not showing a lot of background information in this, except maybe for this one to show that why we should care about the different types of cells that are in adipose tissue. Of course, you have the adipocytes, that are, nice and circular in the lean individual. You have stem cells or pre-adipocytes, here fibroblastic form. You have different immune cells, green referring to M2, red referring to M1. You have a variety of T cells also. Though for inflammation, we have focused primarily on the role of macrophages. And then the profile of adipokines, chemokines that are produced more like anti-inflammatory, although you also have pro-inflammatory molecules that are produced. But with obesity, of course, you have an expansion of the adipocytes. So you have hypertrophy, and you have also remodeling of adipose tissue with, of course, more in one type pro-inflammatory macrophages that infiltrate the adipose tissue and that contribute to the dysfunction, metabolic dysfunction that is in part through secretion of these products into the bloodstream and in other tissues. Let me see if I can. Okay, here, that's good. So we're interested in how we can use dietary factors. I'm going to talk about, fish oil, but we look at others, genetic manipulations, not of humans, but of animals, to understand, maybe some specific genes or proteins that contribute to obesity, pharmacological factors. We primarily have focused, at least for the purpose of this presentation, on inhibitors of the renin, angiotensin, aldosterone system. And then our work has actually moved into the aging and Alzheimer's area. We'll get to that in shortly. So to give you an idea, in case you have additional questions or you have interest in some of the work that we're doing, of course, the central part of it, we're very adipocentric. We've been for many years in my lab. So we look at the expansion of adipose tissue, the development of adipose tissue with the risk differentiation, hypertrophy, hyperplasia, and of course, the inflammation that accompanies obesity and how that can contribute to metabolic diseases in general. And we're interested in understanding not only how the obesity leads to metabolic syndrome, but we're also looking at now how obesity can exacerbate Alzheimer's, breast cancer. The breast cancer work has been done mostly in cells. So we look at how fat cells interact with cancer cells. We're also looking at the renin-angiotensin system and its role in breast cancer. So the endocrine system, we have focused primarily on the angiotensinogen or angiotensin system. And we're using a variety of models, cells, hepatocytes, primary adipocytes, and also, cell lines. We have also developed a really nice model aligned with this interesting aging C. elegans. So we have tools in our lab now to actually expand the aging studies with the C. elegans model. We've been feeding C. elegans tertiary, and interestingly for fish oil, C. elegans actually produces long-chain omega-3 fatty acids. So we use mutants that don't produce that. It ends up being actually a fascinating system. Hopefully next time we'll have data to show on that. And then mostly through collaborations, I don't do any clinical studies, but through collaborations with the bariatric surgeons and other clinicians, we've acquired tissue, specimens, blood, and fat tissue to study. And in terms of dietary components, we're really interested in fish oil. I will show some of the work there. Tertiary, we just published a study on curcumin, and also we looked at vitamin E derivative. And then we do have a industry-funded project for someone who is interested. It was an interesting experience because I never gone to industry, but recently the opportunity came, and I'm glad actually we took it, but it took a lot of negotiation to make sure we do our science. I think that's something that is very important. We learned a lot. And then we look at the white fat and the brown fat. So of course, the white fat here and the brown fat, and we've also been interested in browning as well. So overall, we're interested in the mechanisms that link adipocyte inflammation to metabolic diseases, chronic diseases, aging as well, and finding out what are some of the signaling that are involved. So I think you're all familiar with angiotensin system, at least the classical one, that leads to the production of angiotensin 2. So it's very small peptide that binds, usually utilizes primarily two receptors, AT1 and AT2. Can we make this one go? Or let's see, maybe here is, okay. There is a way to make it go away. Okay, let's leave it there for now, and then we'll manage it later. So we have the type 1 receptor and type 2 receptor that are the primary receptors known to mediate angiotensin effect, but it's really the type 1 receptor, G protein coupled receptor, that mediates the vasoconstrictive effects of angiotensin 2, high blood pressure, and aldosterone production, while the AT2 receptor actually has a more vasodilatory anti-inflammatory effect. We have shown many years back that some of the angiotensin effects on adipocytes, like increased fatty acid synthesis, were actually in part mediated by type 2 receptor, not type 1 receptor, but both receptors are expressed in adipocytes. And of course, there are other peptides like NG19, NG17, of course, H2 that became very, very famous due to COVID-19 has its own receptor. NG17 has a mass receptor. And then we've used especially ACE inhibitors. Of course, these are antihypertensive drugs that are currently used, either ACE inhibitors or angiotensin type 1 receptor antagonists or ARB. The story goes back, more than 20 years ago, when actually studies have shown that there is a positive relationship between angiotensinogen mRNA or even circulating angiotensinogen mRNA waist to hip ratio. And actually in mid 1990s, actually, we were one of the first to show that adipose tissue produces angiotensinogen, very, very high amounts of angiotensinogen produced by adipose tissue. And interestingly, it's also regulated by dietary and hormonal factors. And then following that, there were a series of randomized clinical trials that looked into the relationship between renin-angiotensin system with insulin sensitivity or diabetes, or the risk for diabetes, as well as obesity. So we're just listing some examples here, some with ARB, some with angiotensin converting enzyme. But basically, the majority of these studies have showed that RAS inhibition consistently and significantly reduced the incidence of type 2 in these patients. Some of these were, have other cardiovascular disease complications as well. And then, of course, to do mechanistic studies, we need to go to animal models. So along with the story that RAS is, or increased activation of RAS is associated with obesity and metabolic diseases, on the other hand, if you actually inactivate any of these components of the renin-angiotensin system, AGT or angiotensinogen, renin or ACE, we've done AGT inactivation, we've done AGT type or AT2, type 2 receptor inactivation, and others have done type 1 receptor. Most of these actually have shown a common phenotype in these animal models is reduced adiposity, but the most consistent one has been actually reduced insulin resistance, has been shown in these animal models. So it's very interesting. So I don't know, this one is not supposed to be animated. But anyway, so based on that, we developed the hypothesis that during obesity, there is this overactivation of the renin-angiotensin system. And we propose that if we overactivate the renin-angiotensin system, you develop insulin resistance and likely obesity and glucose intolerance, and that when we use pharmacological inhibitors of RAS, this will reverse it and will lead not just to a reduction of the circulating angiotensins, but also to reduction of inflammation and increased anti-inflammatory molecules. And so earlier studies actually using these transgenic mice, and at that time, because that first study was done back in 2012, so like 10 years already. And so at that time, we didn't have all these, fancy tools. So this model was generated using the AP2. And you're all familiar with the fatty acid binding protein AP2 receptor that now we know is not the perfect one because it's also expressed in immune cells. But it did actually reveal some interesting data. So we looked at both males and females. So there was an increase in body weight, and then we looked at the gonadal fat. There was a significant increase when angiotensinogen is overexpressed in adipose tissues in this AP2 promoter. And then when you look at the fat cell side, there was clearly a hypertrophy of adipocytes in those, we call them OVEX, which are the mice that overexpress in angiotensinogen. And then when we did glucose tolerance test, this is, you can see in females. So this is why most of us, as we were discussing earlier with Vicky, we use males to study diet-induced obesity, because especially in the Black 6 model, you can give high-fat diets. They're quite resistant to developing high-fat diet-induced obesity. And so you can see in the females, we don't see really any significant differences, while when you compare the wild type and the transgenic, this is on a low-fat diet. So what this shows that you have increased glucose intolerance on low-fat diet just through overexpression of angiotensinogen. So then we took these two groups of males and put them on a high-fat diet. I'm going the wrong way. On a high-fat diet, and what we saw is basically high-fat diet with or without angiotensinogen is giving you maximal glucose intolerance. So that means angiotensinogen overexpression mimics the effect of high-fat diet. And then when we used captopril ACE inhibitor, it actually reversed that largely reversed that intolerance. And then we sent at that time these mice to Vanderbilt Mass Metabolic Phenotyping Center through collaboration with David Wasserman to see whether there are really any changes in response to insulin or insulin sensitivity or how they handle glucose. And clearly you can see that with lower glucose infusion rate in the transgenic mice, that means they are clearly resistant. And this is also shown by showing that whole body glucose clearance is lower in the transgenic mice. And then what I think is, to me, is most interesting is when you look at insulin-stimulated glucose uptake in various tissues. And you can see here, and start with adipose tissue, there is no difference. And actually, even after you adjust for adiposity, maybe you see an increase, not a decrease. But when you look at muscle tissue or the heart, there is actually a reduction of glucose transport glucose utilization in the transgenic mice. So clearly there is resistance in these animals, and this is on a low-fat diet. So they were not even given a high-fat diet. So that was very interesting. And then when you look at, of course, when we talk about insulin resistance, we think about cytokines that may be altered. So here you can see CP1 going up, and then IL-10 anti-inflammatory cytokine going down. So then we wanted to look more into the mechanism, what receptor is mediating these effects. And so at that time we had shown in adipose tissue that AT2 receptor actually was mediating the changes in lipid metabolism. About the same time we had published actually cell culture data, I got a call from Gerard Ayot. I don't know if you know who he is. He is the father, one of the fathers or parents of adipose tissue biology, and he had used different cell lines of B17. So he called me and I said, we saw the cell culture data we've done, and he was interested in a collaboration to further explore the renin-angiotensin system. And then I circled back to France where I did my graduate studies and started a collaboration through that with various investigators. And what we tried to see whether angiotensin type 2 receptor mediates these effects based on the cell culture studies that we had. And it turns out it was the case. You can see it here, how the adipocyte size are smaller in the AT2 knockout mice. And of course, transgenic mice, as I showed, are hypertrophied. And then when you delete angiotensin 2 or inactivate angiotensin 2 type 2 receptor in these transgenic mice, you see that reverse that phenotype of the adipocyte. So you get larger adipocytes in the transgenics, and then it was reduced once you remove the type 2 receptor. And then I think this was very revealing as well. You can see all these red lines are the transgenic mice that have high VEGF, high TNF-alpha. Almost every cytokine that you measure is very high. But then again, when you compare this to this pink one, that is the transgenic mice that don't have the AT2 receptor, it's totally reversed. So when you look at this, don't you think that these mice have a great metabolic health and metabolic phenotype? Inflammation down, fat cell size smaller, glucose tolerance is better, but look what happened to blood pressure. Actually, my lab had focused more on the metabolic aspects of the renin-angiotensin system, and my collaborators in France were looking at more the kidney and blood pressure regulation. So you can see with the mice that overexpress angiotensinogen, actually 30% of the circulating angiotensinogen coming from adipose tissue, but they have high blood pressure. When you remove AT2, despite a healthy metabolic profile, blood pressure actually goes even more up. So we haven't followed that story, except that what we found in the kidney and many other tissues, AT1 expression, the type 1 receptor actually expression goes up. We think that that's in part what's driving the high blood pressure now that the type 2 receptor is not there. AT1 is activated and driving the blood pressure. I think this is really very interesting. You're separating the cardiovascular effect or the vasoconstriction from the metabolic effect, and glucose intolerance actually was improved. So then following with yours, I was trying to get this funded by NIH. They really like the transgenic mice studies. And then we have the AT2 receptor studies. And now what you need to do is adipose tissue specific knockout mice for AT2 receptor. And of course, it takes time. You know, you think you need the money to do the work first, but so we had to go and do this. So we generated the angiotensin receptor type 2 knockout mice specifically in adipose tissue, and we did not see any major phenotype like weight loss, etc., on the diet-induced obesity. However, we saw significantly reduced inflammation. And studies by Cassis, those are shown in blue, showed that these mice actually develop as the age. Usually mice have increasing blood pressure, especially on a high-fat diet. These are more resistant to age-related increase in blood pressure. So the main effect of angiotensin type 2 receptor in adipocyte at least seem to be primarily this inflammation that is reduced. So we wanted to understand more back to the transgenic mice, what are the mechanisms beside what we've already shown. So we did profiling, everybody's nowadays now to identify global changes, and we keep hitting these pathways, lipid metabolism, inflammation, and ER stress. And we were really very interested in this ER stress pathway. I think if you are familiar with the CHOP and the 3 unfolded protein response that happen when you have ER stress or misfolding, which occurs in obesity and metabolic diseases. So, so we looked at these pathways to see how they're related to angiotensin energy overexpression. Are these some of the mechanisms mediating the effects of angiotensin through inflammation, of course, coming from the inflammatory adipokines that are coming from the fat tissue. So what we did is to check are there changes in ER stress, and sure enough, most of the markers that we measured of ER stress was actually increased in the adipose tissue. Of course, inflammation, I've already showed you that inflammation was increased in the transgenic mice. And so the other study that we did was, okay, would this be prevented by captopril? So if you inhibit the renin-angiotensin system, would that reduce ER stress? And that's actually exactly what we saw. We again used angiotensinogen transgenic mice. And when we gave wild type or transgenic mice captopril, the ER stress was significantly reduced. So there is this crosstalk between inflammation and the ER stress. And of course, captopril also reduced inflammation in these animals. So now how about the knockout mice? So I showed that the knockout mice on high-fat diet-induced obesity, they were resistant to, there was reduced inflammation. So ER stress was up with overactivation of angiotensinogen, but it did not go up. At least why it did not reduce ER stress, but not having angiotensinogen actually prevented that increase in ER stress. So then we followed up with mechanistic studies. This is part of the work that Kalhara has done using either captopril or using ARB, telmisartan. And what we found when we treat adipocytes, so we used human adipocytes, mesenchymal stem cells, and we also used 3T3 in one adipocytes, consistent data showing that angiotensin 2 increases ER stress. But when we use the AT inhibitor, it was reversed. Especially, actually, we saw reversal with IT1 receptor. Of course, for the inflammation, the same thing. Several markers of inflammation and up. And then with both the AT1 inhibitor, we didn't see as many changes with the AT2 receptor inhibitor. It was mostly the captopril and the AC inhibitor. And then we actually at the same time we did the profiling, we did microRNA and RNA-seq profiling to identify specific target pathways. And we have identified some microRNAs that actually have been previously reported to be related to changes in ER stress, microRNA 30, 708, and 143. So they have all been shown previously microRNA 30 to be downregulated. in ER stress, while 708 and 43 was upregulated. So in transgenic mice, this really was correlated very well with the change with the overexpression of angiotensinogen. And we got interested into this specific microRNA, which is 690, because there was a significant change in that. And then this microRNA actually hit, I think you all know that microRNAs act as silencers by inhibiting, either degrading or inhibiting translation of specific target proteins or genes. And in this case, we found that 690 was actually targeting these interleukin pathways and the MAP kinase pathway. So we focused mostly on this MAP kinase pathway because of its known role in diabetes, et cetera. So we have done, at this point, actually, Kalhara has done additional studies with microRNA mimics and inhibitors. And this is first is to look at if microRNA 690 is actually expressed in adipocytes, and we show in the transgenic mice there was significant upregulation of microRNA 690. And again, when we treat cells with the type 1 receptor antagonist, I think I feel like it's hot here, there was a downregulation of this microRNA 190. So what he went on to do is to use microRNA mimics and inhibitors, because again, even if you see changes in the microRNAs, you still need to confirm that using cell culture with mimics and inhibitors. So I'm going to go a little bit fast over this one to show specifically that while we generally, and I think this is an advice to students, to not just do gene expression, you have to do protein expression, because often, especially for these signaling molecules, you may not see changes at the gene level, but at the RNA, at the gene level, but at the protein level, you would. So when we use 690 RNA mimics, it did inhibit P38 and also P65, so inflammation and the MAP kinase. And this was shown, here as a quantification. So to summarize this part, there are some of the data that I have not, shown. I wanted to show more of the most recent ones. So we have shown that angiotensin 2, of course, RAS is activated, and both angiotensin 2 and angiotensinogen is high in obesity and insulin resistance. It targets adipocyte via these two receptors. Again, I don't know why this one is not animated. And we have shown many years back increased lipogenesis, but we actually looked at more the NF-kappa B pathway as a specific, target of angiotensinogen and angiotensin 2. So in addition, as I just showed you, that the microRNA 690 is targeting the MAP kinase as one of the pathways that is altered by RAS overexpression along with the inflammation. So to summarize this part of our expression of angiotensinogen increases adiposity, activates inflammatory and ER cascades, and that there are benefits of inhibiting the RAS pathway. I mean, we know for hypertension, but also it may be beneficial in metabolic diseases. And this is in part through ER stress. And again, looking at these microRNAs that can target multiple metabolic pathways may be a good approach. So again, this needs to be tested in clinical studies. I don't know. I haven't looked at, I think we were the first to identify this microRNA 690 in relation to angiotensin and obesity, but I think it will be interesting to see if some of these microRNAs are actually biomarkers for, I don't know, hypertension associated obesity. It's worth looking into it. No questions so far? Because I'm going to switch topics to fish oil now. Yeah. Okay. So I will switch topics now to fish oil. So I talked about the endocrine function of adipose tissue. And before getting into this, I do want to mention that we did actually look at whether omega-3 fatty acids regulate the renin-angiotensin system. And what we have published a few years back now, that when you treat adipocytes with the icosapentaenoic acid, there is the reduction of secretion of angiotensinogen from adipocytes. So I think that shows that there may be some benefits of fish oil through the renin-angiotensin system to have some positive metabolic outcome. So we'll talk about the obesity work that we have done on white and brown fat, and then I will end with some very recent data, very exciting data on the Alzheimer's disease. Again, we're in the nutrition department. So I just want to put this to remind you, but I think you've heard probably from Elizabeth and the others, and you're sick of hearing about fatty acids around here. But I do want to point out that it's really this balance. I think that we cannot emphasize enough how our ratio of omega-6 to omega-3 is too high. I'm not saying some people think that we should remove omega-6 from the diet. No, they're essential, right? We know that, but it's the ratio we consume, maybe more than we need omega-6, less omega-3. And it's very important because they compete for incorporation into phospholipid, they compete for producing their own metabolites. And the second point I want to make is that you cannot just increase your very long chain fatty acids by consuming alpha-linolenic acid, because the conversion is not really that simple and that efficient. So you do need very long chain fatty acids in the diet, such as EPA and DHA, to achieve those benefits. And what we're learning now is some of these metabolites. Actually, recently we found that 8-HIP, one of the metabolites of EPA, is even more potent at even lower doses in reducing inflammation. So in addition to these long chain fatty acid, EPA and DHA, we have to pay more attention to resolving and protecting the HIPs, the different metabolites and derivatives that have their own mechanism of action. So again, I want to bring you back to some earlier studies that actually led to where we are now. So we fed mice a high-fat, low-fat diet. This is the red line, or a high-fat diet. There is a blue and green line together here, but it's a high-fat diet, 45% calories from fat. And we saw that, of course, high-fat diet increases weight gain. And we see also, this is glucose tolerance test, we see that the mice that were fed high-fat diet, clear glucose, don't clear glucose as well, while the low-fat diet in red here clear it, better. And in addition, we also gave mice a high-fat diet, but that was enriched in EPA. So this is ethyl ester EPA. So it's almost like 99% pure EPA that we gave at that time. And so what we saw is, compared to this high-fat diet, the high-fat diet enriched with EPA significantly reduced body weight. But I think what was more impressive is, despite this higher body weight than the low-fat diet, you can see that the glucose clearance is almost similar. So this is, we're talking about 45% calories from fat, 10% calories from fat. And then we have the green line here. What we did here is we fed mice high-fat diet, and then we switched them to high-fat diet with EPA to see if EPA can reverse the diet-induced obese effects. As opposed to this one, this group has been fed with EPA along with the high-fat diet for the whole time. And what we see is that there is some, improvements, small improvements of glucose tolerance, despite the lack of, changes in body weight. And then we also see insulin was brought down either by the preventive diet or even short-time feeding of the EPA to the mice. I'd like to see histology pictures because, they tell you more. So this is a low-fat diet and high-fat diet fed the animal. And you can see that both of the EPA groups, whether the EPA was fed the whole time or whether the EPA was fed just for 6 weeks after inducing high fat, and this is independent of body weight. Without any change of body weight, you can see that, you don't have much lipid accumulation with EPA. I think we need to work towards having EPA prescribed for fatty liver once we verify it in humans. I think it has some very promising effect. And of course, we looked at inflammation. Of course, we have those crown-like structures with high-fat feeding, MCP1, and also this is just quantification of the macrophage area goes up. And then again, with the EPA, whether it's a prevention diet, the whole 12 weeks versus just the 6 weeks, you have significant reduction of inflammation. And so I want to summarize here these studies before moving on to what happens in the brown fat. So we have shown that there is a reduction in adipocyte size with the fish oil feeding. There was increased fat oxidation both in white and brown fat, and there was a reduction in inflammation. We have also done some studies with the lipid mediators, and we showed that fish oil reduces leukotriene B in these animals. And we have some studies by a fellow from Brazil who was funded by FAPESP to look at the role of lipid mediators and whether, EPA or fish oil through the reduction of leukotrienes may be actually good mediator to reduce type 1 diabetes as well. So this was the interesting data from the student I mentioned earlier, Mandana. She's now doing actually a postdoc in cardiology at UT Southwestern. So I was really not interested in brown fat. I was just interested in white adipose tissue, fat cell size or hypertrophy and inflammation, but she really wanted to look at brown fat. And so when she did Western blot, she showed that in the EPA, especially the prevention group, there was increased UCP1 protein. And then I said, okay, let's look for browning. We could not really see browning. And as Vicky reminded me this morning, that's because we use males, because later on when you use females, we actually could see browning. At the time, of course, this study was done with the purpose of inducing inflammation and insulin resistance. So we looked at males that really developed the phenotype, but later on when we looked at actually females, we could see the browning of the adipose tissue. So based on this, she went on to measure several other markers of thermogenesis, PGC1, PRDM16, and we published a couple of years ago these data that summarizing that really fish oil induces this thermogenic program, both in males and females in brown adipose tissue. So based on that, actually, this gave the idea for the whole NIH grant that was funded that the effect of fish oil was mediated by uncoupling protein 1. So what we did is to take uncoupling protein 1 deficient mice, so UCP1 knockout mice, feed them fish oil, and the proposed hypothesis is they're not going to respond to fish oil because the UCP1 is not there. Well, it turns out that we were wrong. So I want to show also these pictures. Let me just back up here. When we used the UCP1 mice, of course, we went to the literature that was done. There was some work by Nedergaard and Barbara Cannon in Netherlands that have shown that when you take the UCP1 deficient mice and raise them, maintain them at ambient temperature, they don't gain significant weight. But when you put them in high fat diet and in a thermoneutral environment, 28 to 30 degrees, they're going to develop obesity. So we first decided to go with that approach to take these mice and put both wild type and knockout mice, UCP1 knockout mice at thermoneutrality at 28 to 30 degrees, feed them high fat diet and see what happens. And this is what happened to their brown fat, both high fat, wild type, or knockout high fat. You can see this doesn't look like a brown adipose tissue, especially the knockout mice. Actually, the brown adipose tissue looks just like white adipose tissue. But given fish oil to both of them, significantly reduced adiposity. Actually, in the wild type mice, we see better improvement at ambient temperature than at thermoneutrality, but still we have improvements. And what was interesting actually about these mice is our hypothesis was totally wrong, because when we took the knockout mice and gave them fish oil, they showed, I'm not showing the data, but they showed exactly what we saw in the wild type mice. So they responded by improving glucose clearance, by reducing anemia, and this is it. And the brown adipose tissue, actually, it seemed like it didn't matter. So we're still looking. If anyone has suggestion, we've gone through what are the mechanisms by which these mice are going to, improve this glucose intolerance. And we saw, of course, the increased adiposity in the high-fat mice, but we have also increased mitochondrial protein. This is in UCP1 deficient mice. We've seen these mice have increased oxygen consumption. However, when we looked at the adipose tissue, the white adipose tissue, while EPA had all these improvements, it increased inflammation. So we don't know, this must be a mechanism that is temperature dependent perhaps. So this is the first time that actually we did not see beneficial effect of fish oil on inflammation. We put these mice at thermoneutrality, and when they are also deficient in knockout mice, despite the improvements in insulin resistance. So that's another mystery. So we're trying actually to identify the mechanism. So they said, okay, it has to be the liver, maybe increased oxidation in the liver. So we measured mitochondrial peroxisomal oxidation, and it is actually significantly increased by 5 to 10 times. It's increased in the liver, and we're doing inactivation of specific targets of peroxisomal oxidation. And so far, even in the adipose specific knockout, it doesn't seem like there is a phenotype there. We did also the mitochondrial glycerophosphate dehydrogenase 2, and we looked at the calcium, fertile cycles. There may be some mechanism. It's not that. So at least we've eliminated what is not, working. But we'd like really to identify what's going on. We have not looked at muscle. I think that may be one area that maybe we should be looking at. So we want to hunt these UCP1 independent mechanisms, and we're looking at lipid mediators because I think they really have a lot to do with the mechanism of action of EPA. And then we started looking at browning, both in males and females. And what we're actually finding interesting is there was increased browning at ambient temperature in UCP knockout mice when they are raised at ambient temperature. The 10 to 100-fold browning. I've never seen this change in, at least at the gene expression level of these markers. How are we doing with time? We're good, okay. Because I have one more set of data to show. So of course, EPA has been controversial, but fish oil has been controversial because the consistent effects on triglyceridemia, almost any species that has been tested for the fish oil or EPA effect has shown a reduction in triglyceride in mice, in domestic animals, and you see it everywhere. Inflammation has been, for the most part, consistent too in humans. There were studies in PBMCs, and it showed reduced inflammation as well. But the effects that we see in improved glucose intolerance, improved insulin resistance have not been consistent in humans. And I was discussing with Elizabeth this morning, I don't know why. I'm very excited about EPA actually, because there are some recent clinical studies the past couple of years showing that EPA is better than fish oil, at least in protecting, maybe reducing MI cases. It's more cardioprotective than the whole fish oil. So maybe the studies hopefully will stimulate more the research on EPA. When we first started this work, our goal was to start with EPA and then move to DHA, but we got so excited about what EPA is doing, we never did that. But I think it's still a worthwhile comparison. We did notice that when we use diets, at one point we could not get the pure EPA or the 80% pure EPA. We used a diet that has DHA. We saw less effects on glucose tolerance or even on inflammation. So there is something that EPA is doing or its metabolites are doing that is mediating all of this. And of course, if studies can be done in males and females in different ethnic groups, and diabetes, I really think that EPA, based at least on the prevention and the reversal treatments that we did, I think that EPA works better before full-blown diabetes. If you have pre-diabetes, maybe hypertriglyceridemia with pre-diabetes, I think you have a better chance. Hopefully in humans, someone needs to do that. You have a better chance of improving actually the insulin resistance. And so the last part of the talk that I want to show actually also related to fish oil. So I highly encourage anyone of you who has an NIH grant to apply for supplements because they're easier to get once you have your grant funded. So we had this grant was looking at fish oil and uncoupled protein one. And then there was a call for funding from the National Institute of Aging for funding researchers who are not Alzheimer's researchers, which I'm not, but who are interested in developing that area. So we had a member of our obesity research institute who is a neuroscientist. And so I approached her and I said, would you be interested? And then we submitted it, and it was one of those rare cases that it gets funded from the first time. And so what we propose to do is to look at an amyloidogenic model, APP model of Alzheimer's. And it has nothing to do with UCP1. That was the main purpose of the parent grant, was to look at fish oil and then coupling protein 1. We just proposed to see, does fish oil protect against obesity, or does it alleviate obesity associated with Alzheimer's? So it's a very simple question. And just briefly, because I was not sure, the background on Alzheimer's, how it is, because I had to learn it myself too. I'm not a neuroscientist. So, excuse me, we're interested in the amyloid precursor protein. And so it's a transmembrane protein. And when it gets cleaved by two enzymes, gamma and alpha secretase, so you get this soluble APP that is not a problem. So as long as it's a soluble APP, there is no aggregation. However, there is another enzyme that is called beta secretase. So when the beta secretase actually cleaves this extraneuronal part of the protein, then it will lead to this amyloid, insoluble amyloid beta, and then this is what aggregates, and this is what creates problems that lead to plaques. On the other hand, you also have totally another pathology that relates to neurofibrillary tangles, to tau protein phosphorylation, that both of these can lead to apoptosis. So these are some of the main factors for Alzheimer's. So as you all know, big controversy about new medication that was approved, this aducanumab, that lowers amyloid plaques. I think many people think that it should not have been maybe approved, but it has been approved for Alzheimer's. So we were excited to look at that. So we wanted to, of course, for us, see whether fish oil is beneficial. And the hypothesis that actually Nessa had when she came, she was interested in aging. Right at that time, this grant hit. I said, go develop, this project, and she did, coming with a clinical background and bring in and developing this animal study. So the idea, of course, we know with obesity there is this inflammation, and she proposed that maybe this can lead to neuroinflammation down the road, and this can exacerbate in this model of APP the plaques, and that can increase the aggregation, and that when we use fish oil, would it help in any of these pathways? So we use this APP, transgenic model that has, that's overexpression of APP and the secretase as well. And there are some mutations, of course, in these genes that have been linked to plaque formation and amyloids. So this model develops Alzheimer's around 6 months of age, and the plaques, and we carried it actually to 10 months. So that's all that we could do at this point. So she had a very ambitious experimental design. This is actually more than what we proposed in the supplement. So low-fat diet, high-fat diet, supplemented with EPA. We had males and females, and for the duration of the 10-month, a battery of metabolic assessments was done, and then our neuroscience collaborator did a series of cognitive assessment that she's still analyzing. And so I wanted to show some very interesting data. I hope that the colors help, but the red is the low-fat diet, and you can see how the profile in females and males is very different. And I will show you what surprised me the most. Of course, in males that we always use, the low-fat separates very nicely from all the high-fat groups, regardless of what they are. And you can see that with the triangles, always the EPA is lower. But look at the females. The highest group is the EPA-fed groups. This was totally unexpected because, of course, we've used males all the time, but also because we haven't done as long of a study. So usually we do shorter studies. So this was very surprising, actually, that the female mice gained more weight on the EPA diet. And this is the transgenic group. So the transgenic group that gained the most. So then look at this, the fat mass. So the fat mass was even more interesting because you look at actually, unlike the body weight, the fat mass clustered very well along the diet, right? So this is the low fat with groups here, transgenic or wild type, and these are all the high fat with EPA. Not huge differences. So where is the body weight coming from? There was increased lean mass in the females. Then when you look at the changes in body weight in the males, it's actually along the fat mass. So it seems like EPA and even the transgene affects differently the males and the females. So here you can see clearly with EPA, there was a change in fat mass in both the transgenic and the other mice. And then we looked at, of course, the profile. This is very complicated statistical analysis. We've spent a lot of time trying to see what's the best way to do this. But I want to point to a few things. Of course, any of the groups. So this is the blue, or is this blue, purple, is the high fat group. The red is the low fat. The green is the EPA groups. And then we have each one of the wild type transgenics, and then within each males and females. We thought that this may be an easy way to present it. If you have other suggestions, let me know. But we can see that leptin was high, right, in most of the high fat groups, and reduced. But you can see again, especially in the males, right, you see more reduction of the leptin, which goes along with the reduction in adiposity. On the other hand, you see all the EPA groups, which we have seen even on a short term. Adiponectin is really one of the mechanisms by which EPA acts. And then you can see that insulin, mostly in the males, is going up, and then it's reduced by that. So this is the most interesting result that I really got excited about as I'm aging as well, is that Moussa looked at gene expression of this amyloid precursor protein in the cortex, and also she looked at amyloid beta 4A in the serum. And what we found here, of course, you expect the transgenics to have more amyloid. There was some baseline because probably the mouse and the humans, but you have increased amyloid here, and that was significantly reduced in the transgenic animals here, especially in the females. We saw a reduction. And here, when you saw the amyloid beta in the serum, actually, you see the same pattern that, again, the EPA seems to really be effective in reducing amyloid. So we're very excited about this. We want to pursue more this analysis, but I'm just going to show IL-1 beta. But when you look at the inflammatory markers in the brain, only in the female, so this is a huge increase in inflammation in the brain. So this is one of them in IL-1 beta. So I'm going to skip this actually because I want to get some questions. So just to conclude, targeting inflammation independent of obesity, whether you reduce obesity or not, I think inflammation, just reducing it is beneficial, whether this is through this endocrine system or through the dietary fish oil. And I think for the Alzheimer's, we need to validate this in other models, because there are Tom models, there are different models of Alzheimer's. So I think we need to look more into this, and we need to dissect more the mechanisms that are involved. But it being EPA, being a dietary intervention, I think, hopefully this can be translated by you all who are doing clinical studies. So with that, I would like to thank you again for having me here today. And sorry to throw all this huge amount of information, but I wanted to share some of the work that is going on. And then if you have any questions, I'd be happy to take them. Yes, and I think it's more the size of the adipocytes per se than the total fat mass. And the reason why I'm saying that, for example, in the group where we fed we call it the reversal group, we fed high fat diet, and then we switched them to induced obesity, we switched them to high fat. Actually, the fat mass didn't change, but when we looked at severity, the fat cells were much smaller. And so I think that it's really the size of the adipocyte and the macrophage infiltration, the amount of crown-like structures that are going to have the response rather than just the fat. So this is what I would call the EPA metabolically healthy mouse than the mouse that was just given the high fat. So it's the inflammation in the immune cells. This is shown by the other in other models. And we have another model that I didn't even talk about where we see the phenotype only in females. I'm getting a little bit more interested in the sex differences with the heat shock proteins. You get different responses, and again we see it's the hypertrophy. It's not the size of the adipocytes. In fact, the angiotensin knockout mice or even the treated might actually have more cells, but the cells are smaller. So it's the size of the adipocyte, the immune cells, macrophages. We haven't even looked at T cells. I know they're important as well, but we've just focused on one aspect. Yeah, thank you. Sure. Oh, thank you. Mhm. Right. Mhm. No, we did not. We did only staining, mitotracker type staining, and there was increase. We did expression, we did protein analysis of the coxes. So we did that. We did that. Yes. Yeah. Mhm. Yeah, yeah. I look forward to it, and yeah, I learned a lot from our discussions today too. Yeah, yeah.
Any more questions?
I have one more question. I'm super happy you got the last data set.
Mhm.
The females, can I say they keep muscle better? They're like, and
We don't know about the quality, just the lean mass is higher. Yeah.
lean mass is higher.
Right.
There's association of Alzheimer and lean mass. Alzheimer,
Mhm.
but the same didn't happen to males. So
Mhm.
do you run any correlation between the pretty exploration right now, but the ability of them to keep a lean mass and cortex could be caused by the fact that you're able to keep lean mass in the female.
Yeah, I see what you're saying. We don't know. What I didn't mention is that the transgenics that have the higher lean mass were also, we used the metabolic cages. I didn't actually show that the TEC, and they were more active as well. So there was more activity, and then our collaborator, she's still analyzing the data, but she did the Morris Water Maze, test, and they actually did more distance as well, especially the female. So I'm not very, but she said from the preliminary analysis. Again, when we had that diet that was mixed with DHA, we did not, it was so small difference. It was abrigado. Yeah. Thank you. Thank you for having me. How about the ones who are there?